Malaria is a mosquito-borne disease, and in this unit we will discuss vector control and entomological surveillance in areas approaching malaria elimination. In Session A of this unit, our focus is on mosquito biology and vector control.
By the end of this session, you should be able to explain the life cycle of mosquito vectors and how different vector control interventions exploit or take advantage of various mosquito behaviors. Explain how the concept of vectorial capacity and receptivity are related. Explain which vector control interventions are recommended for large-scale deployment and which ones are considered supplementary. Explain how insecticide resistance develops and the impact it may have on vector control interventions. And describe the analysis countries should undertake before focalizing vector control.
We learnt in Unit 2 that malaria is transmitted between humans by a mosquito vector. Now we're going to learn about the methods used to control the vector and reduce malaria transmission. But first, let's review the mosquito life cycle. There are about 400 different species of Anopheles mosquitoes, including sibling species. Sibling species are genetically different but look the same. Approximately 60 to 70 species worldwide can transmit malaria. Of these, about 40 are vectors of major importance. The list of known species of Anopheles never stops growing, with the frequent identification of new sibling species. The life cycle of the mosquito has 4 distinct stages. The first is the egg stage. Female mosquitoes lay 50 to 200 eggs at a time in water bodies. Different species of Anopheles mosquitoes have their own preferred water habitats. For example, some prefer small, shallow collections of fresh water, such as paddles and animal hoof prints, whereas others prefer large, open water bodies, including lakes, swamps, and rice fields. Some prefer to lay their eggs where there is sunlight, whereas others prefer shade. After 2 to 3 days, the eggs hatch and larvae emerge. The larvae spend most of their time feeding on algae, bacteria, and other microorganisms in the water. Larvae go through 4 stages of development before they metamorphose into pupae. Pupae are comma-shaped. Pupae do not feed but undergo a radical metamorphosis, after which the adult mosquito emerges. The time between egg and adult varies by species and temperature. The development could be as short as 7 days, but usually takes 10 to 14 days in tropical areas. In general, mosquitoes develop through these 4 stages at a faster rate at higher temperatures. One of the reasons there are more mosquitoes in warmer climates. Adult mosquitoes will feed on nectar, but after mating, females will require a blood meal for the development of eggs. After feeding on blood, the female will rest for a few days while eggs develop. When the eggs are ready, the female mosquito will find a water body to lay her eggs. The female will repeat this cycle of blood feeding and egg laying every 2 to 4 days until she dies. Females can survive for a month, but generally live for 1 to 2 days in nature. Once adults emerge from their aquatic habitats, both males and females forage for sugar for nutrition and energy. Sugar can be obtained from nectars in flowers or from fruit. Although blood is needed for females to be able to develop their eggs, sugar remains an important source of energy for mosquitoes throughout their lifespan. After mating, female mosquitoes will feed on animals or humans when seeking a blood meal to help develop their eggs. Female mosquitoes bite because they need blood to mature their eggs. Mosquitoes produce several batches of eggs during their lifetime, one after each blood meal. This frequent need for blood drives mosquitoes to bite humans and other animals and provides an opportunity for the parasite to reproduce and find new hosts. Some species of Anopheles mosquitoes may prefer to feed on animals, called zoophagy, whereas some have a preference for human blood, anthropophagy. This is not a binary characteristic. Mosquito preferences may change depending on the availability of different blood sources at the time, but there are some species with clear zoophilic or anthropophilic tendencies. Anopheline mosquitoes generally do not feed during the day. Anopheline mosquitoes feed between dusk, when it begins to get dark, and dawn, when it begins to get light. That said, some species may bite during the day in heavily shaded conditions. This means that most bites from Anopheline mosquitoes happen when people are sleeping. When it comes to biting, mosquitoes also have different tendencies. Some mosquitoes prefer to bite indoors, called endophagy, whereas others have a preference for biting outside, exophagy. After biting, mosquitoes need to rest in order to digest the blood they have taken. Some mosquito species prefer to rest inside houses, called endophily, whereas others prefer to rest outdoors, exophily. The resting generally happens during the day after they have spent the night searching for food. It is important to know that mosquitoes don't necessarily take all of their blood in only one bite or from only one person. As much as 15% of mosquitoes take multiple blood meals in one cycle, which exposes them to more potential infectious humans or more humans to infective mosquitoes. The mosquito's behavior and life cycle offer several opportunities to intervene to increase their mortality rate, decrease their ability to reproduce, and limit their contact with humans. Currently, there are two main interventions recommended for large-scale deployment for vector control and two supplementary interventions. There are a number of field trials underway evaluating other vector control interventions, and in the future, WHO may have evidence support recommendations for additional vector control interventions. The first intervention utilizes the mosquito's habit of biting at night. Insecticide-treated nets, or ITNs, protect sleepers from mosquito bites and, as we'll see, also repel, disable, or kill mosquitoes. The second intervention utilizes the mosquito's habit of resting indoors after it feeds. Indoor residual spraying, or IRS, coats the walls, eaves, and ceilings of dwellings with an insecticide that lasts a number of months, with the residual activity depending on the type of insecticide and the material of the wall. The insecticide may repel mosquitoes, but any that land and rest on the wall are likely to be disabled or killed by the insecticide. ITNs and IRS are the two main interventions for vector control. There are two additional interventions that are considered supplementary. First, larval source management, or LSM, targets the immature stages of the mosquito, that is, the eggs, larvae, and pupae. Managing the water bodies in which mosquitoes lay their eggs reduces the number of adult mosquitoes that are produced. LSM is an umbrella term that includes larviciding, habitat modification or manipulation, and biological control. We'll talk about all of these in the next few slides. Second, it is possible to modify houses to reduce the entry of mosquitoes into the home. WHO currently recommends screening windows, ceilings, doors, and eave spaces. In this slide, we have reviewed vector behavior and the way the interventions exploit that behavior to reduce or interrupt malaria transmission. It should also be noted that human behavior also plays a role in vector control, first by determining an individual's level of exposure to vectors mosquitoes, and second by determining how well vector control interventions are deployed or used. We mentioned on a previous slide that there are around 40 Anopheline species that are considered the primary or secondary malaria vectors in an area. We know more about the behaviors of some species than others, and even among the top 40, there are some mosquito species that are more involved in malaria transmission than others. This table highlights the behavioral tendencies of some of the key vectors in Africa, the Americas, and the Asia-Pacific region. It is important to remember that while these tendencies are characteristic of these species, all mosquitoes can exhibit variation depending on local conditions, and the long-term deployment of certain interventions may result in changes to mosquito behavior. Anopheles gambiae is a very important vector in Africa because it has a very high preference for humans and bites indoors. In Asia, however, Anopheles diarrhea can be very important. in areas where people spend a lot of time outdoors, for example logging or foraging. In the Americas, Anopheles albuminus is also an outdoor biting mosquito that may feed more on animals if they are more available. Anopheles stevensi is an urban vector from Asia that has now invaded the Horn of Africa. This species appears to prefer animal blood but does bite humans and prefers to bite and rest indoors. Take a look at this table and try to identify mosquitoes in your region.
Now that we understand the life cycle of malaria vectors, it is also important to understand how the characteristics of vectors affect the rate of transmission of malaria. Malariologists have developed a parameter called vectorial capacity that approximates the number of new infections arising per day from all the bites on one infectious human, with certain assumptions. This concept of vectorial capacity summarizes all of the important features of a population of malaria vectors and their environment at a given point in time. Let's talk through the factors that affect how many secondary infections are generated from a primary infection. In this slide, the small boy is infected with malaria and is carrying gameticides, the sexual stage of the parasite. In any given day, he will be bitten by some number of Anopheles mosquitoes. This depends on the abundance of mosquitoes, represented by M, and the daily blood feeding rate, represented by A. The daily mosquito blood feeding rate, A, depends on the time between blood meals and the propensity of the vector species to feed on humans. It is assumed that each mosquito that feeds on the boy will acquire at least one male and one female gametocide. The number of days required for the extrinsic incubation period, or sporogeny, of the parasite within the mosquito is represented by N. We learned from Unit 2 that the duration of sporogeny depends on the parasite and mosquito species and ambient temperature, but generally it takes from 8 to 10 days. That's a long time for mosquitoes who have relatively short lifespans. The mosquito's daily probability of survival is P. Most mosquitoes will die during sporogeny, and only a few will survive the extrinsic incubation period to become infective. The proportion of mosquitoes that will survive the extrinsic incubation period to have infective sporozoites is represented by P raised to the power of N, or the daily probability of survival raised to the number of days required for sporogeny. The people bitten by mosquitoes that survive the extrinsic incubation period will be infected. This rate is again determined by A, the daily mosquito blood feeding rate. As different vector species will have different behaviors that affect their biting rates and propensity to feed on humans, and even different rates of sporogeny, you can see that vectorial capacity will be different for different vector species at different places and times. The original formula for vectorial capacity, also known as the Ross-McDonald model, is shown here in case of interest. This formula has been useful for understanding how interventions that affect different mosquito behaviors alter the rate of malaria transmission. However, it is very difficult and almost impossible to measure these different parameters in the field. Therefore, vectorial capacity is more useful as a concept than as a measurement of transmission. Receptivity is a construct developed during the Global Malaria Eradication Program that relates to the transmission potential of an ecosystem. The WHO definition is the degree to which an ecosystem in a given area at a given time allows for the transmission of malaria. Receptivity of an area to transmission of malaria is related to the capacity of the vector population in the area to transmit malaria infections. If mosquito density is too low, if there are no suitable larval habitats, if the vector in the area prefers to feed on cattle, or if the daily survival rate is so low that no mosquito survives the extrinsic incubation period, then the capacity of vectors to transmit malaria will be very low. The types of vectors in an area and their vectorial capacity are key components of receptivity. However, note that the definition of receptivity applies to an ecosystem, which is more than just the vector. An ecosystem also includes humans, the parasite, and the climate. So the receptivity of an area is also affected by the human population, the parasite species, and the climate. Let's consider different factors and how they affect receptivity. Receptivity increases with increasing capacity of the vectors in the area to transmit malaria. The more vector mosquitoes bite humans, the higher the level of receptivity. This means that areas with high numbers of efficient vectors will be more receptive. However, the use of effective vector control can reduce receptivity by preventing mosquitoes from biting humans. Receptivity will also increase in warmer and wetter weather. Warmer temperatures speed up the development of the parasite in the mosquito and the growth of mosquitoes, leading to more infective mosquitoes. Wetter weather increases the availability of larval habitats for mosquitoes, which contributes to greater numbers of mosquitoes and a higher level of receptivity. Receptivity will also increase as population immunity declines, with decreasing transmission. If there are fewer people with partial immunity to malaria, they will be more likely to infect mosquitoes, that is, no transmission blocking immunity, and more likely to acquire infection when bitten by an infective mosquito. The level of receptivity varies geographically and over time. Receptivity is a complex concept, and there is no gold standard for measuring receptivity. When we get to entomological surveillance, we will further discuss approaches to measuring receptivity with entomological data. We will also explore receptivity in more depth in the unit on prevention of re-establishment. Please break into small groups based on your country or region of origin. Use these questions to consider the principal malaria vectors in your country and identify their behavioral tendencies. Next, consider the behaviors and activities of the people living in your area. What behaviors of the mosquito vectors in your area are likely to put people most at risk for malaria?
Let's talk about the two methods recommended by WHO for large-scale deployment to prevent, control, and eliminate malaria.
Mosquito nets were used back in the days of Cleopatra to protect sleepers from insects biting at night and disturbing sleep, and their use has continued through modern times as personal protection against nuisance biters. In the 1980s, bed nets were dipped in permethrin, an insecticide of the pyrethroid class, and trials were undertaken that showed they were protective against malaria. As a result, ITNs became one of the principal interventions against malaria. By sleeping under an ITN, people are physically protected from mosquito bite by the barrier the net provides. The physical barrier of the net prevents mosquitoes from getting close to the person sleeping underneath. That said, the net integrity can degrade over time if nets are not used carefully and maintained. The ITN is more than just a physical barrier. It also contains an insecticide that repels, disables, and even kills the mosquitoes that come into contact with the insecticide on the netting material. If there are enough ITNs in an area, the effect of the insecticide can decrease the mosquito population size and reduce the lifespan of mosquitoes. With fewer mosquitoes to transmit malaria, and many of them dying before the parasite has developed, ITNs can protect the whole community against malaria. This is known as the community or mass effect. As a result, the impact of ITNs arises from a combination of personal protection from biting mosquitoes and a community effect that arises from an overall reduction of the mosquito population. This community effect can benefit even those who do not use ITNs, because there will be fewer disease-causing mosquitoes around to bite them. Currently, the class of insecticides used on ITNs is exclusively the pyrethroid class. Pyrethroids include insecticides such as deltamethrin, alpha-cypermethrin and permethrin. In the early 2000s, manufacturers found a way to make the insecticide in the net last longer. This gave rise to long-lasting ITNs that could kill mosquitoes for up to 3 years. There are also insecticides retreatment kits that can be used to increase the duration of the insecticide on the mosquito net. However, as we'll learn in a few slides, the use of a single class of insecticides, that is the pyrethroids, on ITNs has accelerated the development of insecticide resistance amongst malaria vectors. As a result, new ITNs are being developed with different types of insecticides or other chemicals that can kill or disable mosquitoes. There are also ITNs under development that contain 2 active ingredients. These are called dual AI nets. WHO now recommends 2 different types of ITNs. One that contains a pyrethroid insecticide and a synergistic chemical called piperonyl butoxide, PBO, for areas with pyrethroid resistance, and one with just a pyrethroid for areas without insecticide resistance. A list of pre-qualified ITNs can be found on the WHO website.
WHO recommends that countries use a combination of mass and continuous distribution of ITNs through multiple channels to achieve and maintain optimal ITN coverage and use. Optimal coverage refers to providing populations at risk of malaria with access to the intervention, coupled with health promotion to maximize use and ensuring timely replacement. In practice, this often means calculating the number of ITNs that would be required for everyone to have access, with recognition that system inefficiencies and use patterns might not result in 100% coverage. Mass campaigns, either door-to-door or at a central point within a village, typically occur every 3 years. To ensure sufficient coverage, programs generally need to allocate 1 net for every 2 persons in a household, with an extra net in case there is an odd number. In order to have enough ITNs, the population is divided by 1.8 to determine the number of ITNs to be distributed. In addition to mass campaigns, ITNs can be distributed continuously through antenatal care and immunization clinics. Pregnant women are provided with an extra ITN to ensure protection during pregnancy and to cover the newborn after birth. Provision of ITNs at immunization clinics can also ensure that children are completely covered. Other channels for continuous distribution should also be considered, including schools, non-governmental organizations, and faith-based agencies. These continuous distribution channels can help ensure sufficient population coverage with ITNs during the period between mass campaigns. One approach that is not recommended for ITN distribution is top-up campaigns. In between mass campaigns, if continuous distribution channels aren't working well, households may not have enough ITNs to cover their members. Top-up campaigns try to calculate the need for ITNs at a household level and supply that amount of ITNs to a given household. This approach is prone to miscalculations, requires a significant amount of effort, and ultimately does not appear to be effective at maintaining coverage. A country's efforts are better directed towards well-organized continuous distribution and mass campaigns. However, in elimination settings, as part of focus investigations or other small area investigations, if ITN coverage is considered inadequate, top-up campaigns may be needed. In countries' national strategic plans, the mechanisms for ensuring adequate coverage of the populations designated for ITNs should be clear. Mass campaigns require significant micro-planning to ensure that the correct number of ITNs reaches the populations at risk and that there are sufficient ITNs to cover at a rate of 1 ITN per 2 persons. The issues of ITNs does not end with the issuance of nets. The population needs to understand how to hang and wash their nets and when and how to dispose of them. Public health communication campaigns can help explain how to use and maintain ITNs and dispose of them safely once new ITNs are provided. ITNs are likely to be most effective when malaria vectors bite mostly at night after people have retired to sleep, because that's when people will be sleeping under ITNs. ITNs can be used outside if they are hung up, so they can still be effective in areas where people sleep outdoors. ITNs will be most effective in areas where vectors remain susceptible to pyrethroids. However, when resistance is present, ITNs with PBO should be procured. Finally, ITNs that are pre-qualified by WHO are likely to be the most effective ITNs available, as WHO ensures that they are safe, effective, and manufactured to high-quality standards.
IRS is the coating of interior walls, eaves, and ceilings of a house or other structure, such as domestic animal shelters, with an insecticide. The insecticide has residual efficacy, meaning it lasts for several months. If you remember back to the slides on mosquito behavior, many mosquitoes will enter houses and rest on the walls during the daytime to digest their blood meal. If the wall that they rest on has been sprayed with an insecticide, then the mosquito will be disabled or killed. Mosquitoes might even be deterred from entering houses with walls sprayed by IRS and driven away. Unlike ITNs, IRS does not provide a barrier between people and mosquitoes. Mosquitoes are affected when they encounter a sprayed surface, which is usually only after they have already bitten. So, IRS does not provide direct personal protection from mosquito bites, but instead reduces the mosquito's lifespan, making it less likely for the mosquito to transmit malaria. When coverage is high enough, IRS decreases the size of the mosquito population and provides an overall community benefit. There are many different types of insecticides that can be used for IRS among the class of pyrethroids, carbamates, organophosphates, and neonicotinoids. WHO has pre-qualified specific products that meet the essential criteria for IRS, including entomological efficacy, safety, and quality. Rotation of insecticides is critical to maintain their effectiveness by reducing the potential for development of resistance.
Proactive IRS is generally deployed campaign style once or twice a year. The number of rounds and timing of the campaigns depends on the seasonality of vectors and therefore the malaria season in a country. Best practice is to schedule the completion of the campaign to coincide with the start of the build-up of vector populations just before the onset of the peak transmission season. This ensures fresh deposits of insecticides, as mosquitoes would otherwise be increasing in numbers. The number of rounds will depend on available resources and whether one or two applications are required for the residual life of the insecticide to cover the transmission season. Timely and good quality delivery of IRS operations depends on strong program leadership and a well-monitored management system. Successful IRS campaigns require a high level of political commitment, dedicated human, logistics, transport and financial resources, and adequate organizational and planning capacity. Community awareness and support are other critical factors that influence the effectiveness of IRS programs. WHO has a practical manual available to help with the development and management of IRS campaigns. Reactive IRS is deployed around a confirmed index case of malaria. Reactive IRS is currently recommended in areas approaching elimination or post-elimination settings preventing re-establishment of transmission in the houses of confirmed cases and their neighbours to prevent or reduce transmission of malaria. Reactive IRS is triggered by the detection and notification of a confirmed malaria case, the index case, from a health facility or a community health worker. During the case investigation, the likely location of infection of the index case is identified. During reactive IRS, houses located within a predefined radius from the likely location of infection, usually the index case's household, are sprayed with a residual insecticide. In areas approaching elimination or in post-elimination settings where proactive IRS is already carried out, programs consider switching to reactive IRS only depending on the level of transmission or the receptivity of the area. Programs considering adding reactive IRS on top of proactive IRS should balance the potential added benefit with the increased costs and risk of development of insecticide resistance. In areas approaching elimination or in post-elimination settings where no IRS is carried out, initiating reactive IRS may be beneficial, depending on whether IRS is a suitable vector control intervention. However, if the index infection was imported and the household is not located in a receptive area, there may be no added benefit from reactive IRS. IRS is likely to be most effective when mosquito vectors mostly feed and rest indoors because that's where the mosquitoes will encounter the insecticide. IRS is also most effective when the population sleeps indoors, as there's little impact of IRS on outdoor biting mosquitoes unless they also rest indoors. If proactive IRS is used, the malaria transmission pattern should allow for the population to be protected by IRS with just 1 or 2 rounds per year. IRS is not suitable as an intervention if the structures are not suitable for spraying. This could occur if structures are tense or do not have walls. Finally, the insecticides used in IRS must be able to kill mosquitoes, so vector susceptibility to the insecticide is paramount. As with ITNs, insecticides should be pre-qualified by WHO so that they are safe, effective, and manufactured according to high-quality standards. Countries may be interested in doing whatever it takes to eliminate malaria, and that could mean using both ITNs and IRS in the same areas. However, there is no clear evidence that co-deploying these two interventions leads to better vector control. WHO recommends that effort and resources would be better spent by ensuring good quality implementation of one of these interventions rather than co-deploying the two. One exception to this recommendation is that once high-quality coverage with one core vector control intervention has been achieved, programs could consider deploying the other core intervention to prevent, manage, and mitigate insecticide resistance. If ITNs and IRS are co-deployed, IRS should be carried out with a non-pyrethroid insecticide to prevent the development of resistance. The decision to deploy a second core intervention should only be taken after conducting a prioritization analysis across other malaria interventions, not just vector control, to ensure maximum impact of resources. There are currently 2 vector control interventions recommended by WHO that are considered supplementary to ITNs or IRS.
LSM is an umbrella term that includes 4 different strategies. Larviciding, the regular application of biological or chemical insecticides to water bodies. Biological control, the introduction of natural predators into water bodies. Habitat modification, a permanent alteration to the environment, such as drainage canals. And habitat manipulation, a recurring activity such as flushing of streams. Because all LSM interventions attack the immature stage of the mosquito, they only reduce the size of vector populations. LSM has no impact on the adult mosquitoes that transmit disease and does not provide a barrier between mosquitoes and people. As a result, it does not provide direct personal protection and is generally not as effective an intervention as either ITNs or IRS. In addition, there are limited data on the efficacy or effectiveness against malaria of the 4 strategies that make up LSM. The intervention with adequate evidence of a positive benefit is larviciding. WHO recommends that larviciding could be a supplementary intervention in areas where coverage of either ITNs or IRS is already high and where the water bodies used by mosquitoes to deposit their eggs are few, fixed, that is located, don't change within and between seasons, and findable. The location likely to be most suitable for larviciding are urban areas and arid regions. WHO has pre-qualified a number of larviciding products. Many eliminating countries find it challenging to implement ITNs and IRS in urbanized areas or where mosquito densities are low, and as a result, these interventions may be less acceptable to the population. In such areas, various approaches to LSM might be helpful in reducing mosquito density, but should be conducted with effective monitoring and supervision programs. Some eliminating programs may implement reactive larviciding in response to a confirmed case. However, there have been no research studies evaluating the impact of this strategy.
Recently, in 2021, WHO reviewed evidence on the impact of housing modifications to reduce malaria transmission. Housing modifications are defined as any structural changes pre or post construction of a house that prevents the entry of mosquitoes or decreases exposure of inhabitants to vectors. Housing can be modified when first constructed or through modifications to existing house designs. Housing modifications are likely to be most effective against mosquitoes that are indoor biting and indoor resting. After reviewing research evidence, WHO now recommends the use of screens on windows, ceilings, doors, or eave spaces in houses to prevent malaria. Programs interested in house screening should consider how the intervention will be delivered or maintained, whether the structure and condition of residential houses allow for the installation of screens, and the feasibility and resources needed for implementation if deployed on a large scale. Other housing modifications will be reviewed once there is sufficient epidemiological data. Now that we know which vector control approaches are recommended by WHO, we will review how these different vector control strategies alter vectorial capacity. If you remember from earlier in this unit, vectorial capacity is the number of new infections arising per day from all the mosquito bites on one infectious human. It is determined by the abundance of vectors, their daily blood feeding rate, the length of time it takes for the parasite to develop within the mosquito, the lifespan of the mosquito, and its blood feeding rate. As we learnt earlier, ITNs provide a physical barrier between mosquitoes and people sleeping under the ITN and contain an insecticide that kills mosquitoes. ITNs target mosquitoes that are attempting to feed on humans. The barrier effect of ITNs reduces mosquitoes' ability to feed on humans, reducing A, the daily blood feeding rate. This parameter appears twice in the vectorial capacity equation, indicating that it has substantial impact on transmission. The insecticidal activity of the ITNs reduces the mosquito's probability of survival, P. Vectorial capacity is also particularly affected by this parameter. Over time, the higher mortality rate in the mosquitoes will reduce their overall abundance, M. ITNs affect vectorial capacity in multiple ways, which is one of the reasons they have been an important malaria prevention tool. IRS targets indoor resting mosquitoes. Typically, mosquitoes rest only after they take a blood meal. As a result, IRS has no impact on the blood feeding rate, A. IRS kills the resting mosquitoes, which reduces the daily probability of mosquito survival, P. Over time, the higher mortality rate in the mosquitoes will reduce their overall abundance, M. Although IRS doesn't affect the daily blood feeding rate, it can have a substantial impact on vectorial capacity by decreasing the mosquito lifespan and mosquito density in the community. Larviciding is the regular application of biological or chemical insecticides to water bodies to kill mosquito larvae. Larviciding has no impact on the blood feeding or survival rates of adult mosquitoes. However, killing larvae reduces the size of the adult mosquito population and therefore vector abundance, M. Because larviciding affects only one of the parameters of vectorial capacity, it has a more limited impact than either ITNs or IRS. The final supplementary vector control intervention currently recommended by WHO is the use of screens on windows, ceilings, doors, or eave spaces in houses. Screening prevents the entry of indoor resting or indoor feeding mosquitoes, which reduces their daily blood feeding rate. There is currently insufficient evidence to say whether reducing mosquito entry into houses has an overall effect on the mosquito population size in areas where vector mosquitoes prefer to feed on humans. Most, but not all, vector control interventions rely on insecticides, and the development of resistance to insecticides is therefore a threat to their continued effectiveness.
Insecticide resistance is defined as the ability of mosquitoes to survive the effect of an insecticide. There are several types of resistance, but the type that is of most concern to malaria elimination is physiological resistance. Resistance arises after repeat exposure to an insecticide.
First slide, initial susceptible population of mosquitoes. Susceptible mosquitoes are shown in yellow. A few mosquitoes, shown in red, carry mutations that confer resistance to the insecticide. Slide 2, population after exposure to insecticide. After insecticide has been applied, most of the susceptible mosquitoes, shown in yellow, have died. However, all of the resistant mosquitoes, shown in red, have survived. Slide 3, population recovers. The mosquitoes that were not killed by the insecticide reproduce. Some susceptible mosquitoes from outside the area may migrate in. Slide 4, subsequent insecticide exposure. After another application of insecticide, most of the remaining susceptible mosquitoes have been killed, while the resistant mosquitoes have survived. Slide 5, resistant population. Over time, the proportion of the mosquito population that is resistant will become very high, and the effectiveness of vector control is reduced.
Mosquitoes are considered resistant to insecticides when they can survive exposure to a standard dose of insecticide. WHO considers insecticide resistance to be confirmed when fewer than 90% of mosquitoes are killed in a standard assay. When a susceptible population is tested with the standard assay, 98% or more should be killed. When the result is between 90 and 97%, insecticide resistance is possible and must be confirmed with more tests. It is important to note that mosquitoes can evolve different resistance mechanisms. We will talk about the two main types of resistance evolved by malaria vectors in the next few slides. Resistance to the pyrethroid insecticides used in ITNs is widespread through malarious areas and is increasing. This map comes from the WHO Malaria Threats Map and shows the results of insecticide-resistant assays for pyrethroid insecticides in malaria endemic areas. Data are available from 2010 to date. This slide shows survey results from 2017 to present. The red dots show areas with confirmed resistance, the yellow dots show areas with possible resistance, and the green dots show areas with susceptible populations. This map also provides data on another class of insecticides, besides pyrethroids. However, because, until recently, ITNs only contained pyrethroids, the global malaria community has been particularly concerned about pyrethroid resistance. The link to the threats map is shown on the slide and should be consulted for the most up-to-date information. In areas with pyrethroid resistance, pyrethroid ITNs are more effective if they contain a chemical, piperonyl butoxide, PBO, that blocks the resistance mechanisms in mosquitoes and renders them susceptible again. We will talk about this new type of ITN on the next slide. As just mentioned, WHO now recommends that a new type of ITN be used in areas with insecticide resistance. These ITNs still include a pyrethroid insecticide, but have an additional chemical called PBO that can make mosquitoes susceptible to the insecticide again. PBO is not an insecticide, but another chemical called a synergist that blocks the resistance mechanism that the mosquito has developed. This type of net is called a pyrethroid PBO net. By adding the PBO to ITNs, researchers have shown that the nets are more effective than standard ITNs in areas where metabolic pyrethroid resistance is present. WHO recommends that pyrethroid PBO nets be used where the malaria vectors are resistant to pyrethroids. Regular ITNs can be used in all other areas. All ITNs for which an impact on malaria has been demonstrated contain pyrethroids. However, in the near future, it is likely that ITNs with more than one active ingredient will be approved by WHO as the world continues to combat the threat from insecticide resistance.
Development of insecticide resistance is a problem for IRS in a similar way that it is for ITNs. However, while currently there is only one class of insecticide that can be used for ITNs, namely pyrethroids, IRS has the advantage of having additional classes of insecticide with WHO pre-qualified products available: pyrethroids, deltamethrin, lambda-cyhalothrin, alpha-cypermethrin, etc., organophosphates, malathion, phenyltrethion, pyrimidphosmethyl, carbamates, bendiacarb propoxyl, and neonicotinoids, clothianidin. Overuse of the same class of insecticide, whether through use of ITNs or IRS, can lead to development of resistance. Therefore, WHO recommends that vector control programs avoid using a single class of insecticide everywhere or over consecutive years. Instead, programs should use different insecticide-resistant management interventions. In particular, this means that countries should avoid using IRS with pyrethroids in areas where pyrethroid ITNs are deployed. In a few slides, we will talk more about if and when countries might choose to deploy ITNs and IRS at the same time. However, the most important issue is to never deploy two vector control approaches that contain the same class of insecticide at the same time and place. IRS with a WHO pre-qualified product is recommended for large-scale deployment in most malaria endemic locations. While some countries continue to use DDT for IRS, it should be noted that no DDT-containing product has been pre-qualified by WHO and none is under assessment. This means that there has been no comprehensive assessment, based on manufacturers' data, of the efficacy, safety, and quality of DDT for vector control, and no inspection of manufacturing sites. DDT has been classed as a persistent organic pollutant, and its use is restricted by the Stockholm Convention on Persistent Organic Pollutants. In line with the Stockholm Convention, WHO supports the global goal of reducing and eventually eliminating the use of DDT, while minimizing the burden of vector-borne diseases. In deciding whether to use DDT, a national program should conduct a detailed assessment that explores the potential use of alternatives. If, at the end of such a process, DDT is selected for IRS, this choice should be well justified, and DDT must be used in accordance with the conditions set out in the Stockholm Convention.
There are challenges and opportunities for each of the vector control interventions recommended by WHO. Let's divide into small groups and discuss this question. What do you consider to be the challenges and opportunities of each of the WHO recommended interventions from your program's perspective? Consider the effectiveness, suitability, financial, logistical, environmental, as well as the community acceptability of each intervention.
Outdoor transmission may become increasingly important as elimination nears because good vector control generally reduces the risk of malaria from indoor biting and indoor resting mosquitoes. But outdoor resting and outdoor biting mosquitoes are less affected by current vector control interventions. While LSM may help to reduce the abundance of outdoor resting and outdoor biting mosquitoes, there are other potential interventions that target outdoor transmission under development and evaluation. These include interventions that target sugar feeding, such as attractive targeted sugar baits. Communities should understand the risk of malaria transmission outdoors and be encouraged to avoid spending time outside during peak mosquito biting time if possible, unless they are sleeping under a treated net. Where it's not possible to avoid being outdoors at night, wearing long-sleeve shirts and trousers can help reduce mosquito bites. Topical repellents and insecticide-treated clothing have been proposed as potential methods for malaria prevention outdoors. These interventions do reduce biting and could provide individual protection from malaria vectors. However, the studies of topical repellents and insecticide-treated clothing have not shown these interventions to be effective in reducing malaria in communities as a whole due to a limited compliance. Repellents, for example, need to be reapplied quite frequently to remain effective. The malaria community is continuing to research new and innovative methods to tackle the important problem of outdoor transmission, and there is hope that new interventions will soon be available. National programs are almost always very concerned about how or whether to continue vector control when elimination is close to being achieved. People living in areas that used to be but are no longer malarious may not want to use vector control any longer than they have to, and ministries of health may want to redeploy their limited health budgets for other interventions. However, it is very important that countries understand that withdrawal of vector control before elimination has been achieved has frequently been associated with resurgences of transmission. There have been numerous reports of countries getting close to elimination of malaria and then, because of budgetary issues or believing that elimination was imminent, deciding to scale back their vector control. Almost universally, transmission picked up again very quickly. On this slide, we present two examples. The first, from Sri Lanka, shows how the country came very close to eliminating malaria during the Global Malaria Eradication Programme. In 1963, Sri Lanka had only 6 indigenous cases of malaria. As a result of this success, the program halted most IRS throughout the country. Within months, cases began to resurge, and although spraying was restarted in some effective areas, it took Sri Lanka years to catch up to the resurgence of transmission. On the right, we have an example from the highlands of Madagascar. All IRS was ceased in 1975, and cases began resurging. Other programmatic declines occurred in 1979, and then the increase in cases became exponential. In both of these cases, the withdrawal of vector control was not the only factor that contributed to the resurgence of transmission, but it was an important one. The speed with which transmission will increase after vector control is scaled back or withdrawn will depend on several factors, including the underlying receptivity of the area, the current level of transmission, and the rate of importation. But countries close to elimination should not risk the gains they have made towards elimination by withdrawing vector control at such a critical time. In areas approaching elimination but with ongoing local malaria transmission, vector control interventions should be maintained. They should not be scaled back, meaning coverage decreased, or withdrawn because of the high probability of a resurgence of transmission in the absence of vector control. Optimal coverage of the population at risk with effective malaria vector control should be pursued and maintained. In countries where transmission has been interrupted, programs are urged to maintain vector control during their 3 years of zero indigenous cases before qualifying for certification. If even a single indigenous case occurs during that period, the country will have to restart its certification timeline. That is to say, if a country has gone 35 months without an indigenous case of malaria, but then experiences an outbreak that leads to one or more indigenous cases, the 3-year timeline begins again. To avoid this disappointing situation, vector control should be maintained throughout this period. After certification, vector control should be focalized and continued in areas to prevent re-establishment. The areas at highest risk of re-establishment should be determined based on a detailed analysis that includes an assessment of the receptivity and rate or risk of importation, strength of the disease surveillance system, and capacity for case management and vector control responses. Border areas neighboring higher burden countries are frequently areas of high receptivity and high risk of importation, and should be considered for continued vector control. It is important to know that while space spraying, which is the dispersal of a liquid fog of insecticide into an outdoor area to kill adult insects, is recommended for control of dengue fever in emergency settings or epidemics, it is not an intervention recommended for controlling malaria. There are several reasons for why this intervention is not effective against malaria. First, space spraying is often done during the day when Anopheles mosquitoes are resting. The fog of insecticide is unlikely to penetrate many of their resting sites. Second, there is no residual activity of the insecticide, so it does not persist, meaning it is only effective for a very short period of time and would only affect very few mosquitoes that might be in flight during that brief window. Space spraying is frequently used as a public relations exercise due to its visibility. However, this is expensive and not an efficient use of resources that would be better spent on effective vector control, particularly IRS or ITNs.
In summary, WHO recommends ITNs and IRS for large-scale deployment and reactive IRS at very low levels of transmission. Larviciding and house screening are supplementary vector control measures recommended by WHO. Receptivity of an area to malaria transmission is largely determined by the capacity of malaria vectors to transmit malaria parasites. Some new ITNs include PBO, which makes them more effective than regular ITNs in areas with metabolic resistance to pyrethroids. After elimination and certification, the decision to focalize vector control should be based on a careful assessment.